السلام عليكم. إني الأستاذ الدكتور أنطوان صبري البنا. محاضرتي عنوانها COVID-19 causative virus origin and antiviral drugs and vaccines. Main characteristics of coronaviruses are enveloped RNA viruses. Their size range from 120 to 140 in nanometer, with a spike-like projections on its surface, giving the virus a crown-like shape under the electron microscope. For this, they call it coronaviruses. The envelope is bilayer of lipid membrane acquired through budding from the cytoplasmic membranes. The spike is glycoprotein function in receptor recognition on the cell, and also for cell fusion for entry, and is the species-specific antigen for neutralizing antibody and protection. Spike protein are variable in mutant viruses. Nucleocapsid inside the virus is helical shape surround the nucleic acid, function in a protection of the genome. Coronaviruses have the largest viral RNA genome, range from 27 to 32 kilobase multiplication only in the cytoplasm of the cell. This shows for you the anatomy of the virus. You will see inside the yellowish spiral, which is the nucleocapsid. Inside it is the viral RNA. On the surface, the red peplomers projecting from the envelope are, we call it the S. Glycoprotein is very important part of the virus, and you will see the envelope and the viral membrane with bluish violet coloration. And here the picture taken by electron microscope, negative staining of the coronavirus. Coronaviruses are now under a family which occupy many viruses which infect human and animal. They were classified into 4 genera. The human coronaviruses belong to alpha and beta, and you'll see there are first 4 viruses which are discovered in the 60s, and they cause only a common cold or very mild respiratory infection. Other human coronaviruses which causes severe respiratory syndrome, first your SARS, which causes acute severe respiratory syndrome, and this discovered in China, Guangdong in 2002. In animal food market, bats are the reservoir, and they pass the virus to civet cats to human, and this SARS spread to 26 countries, which result in 8,000 cases, 774 deaths, which is about 9.6% mortality rate. MERS is also causes Middle East respiratory syndrome. MERS first isolated in Saudi Arabia in 2012 from respiratory secretions of a human with severe pneumonia. Bats are the reservoir host, and camels harbor and shed MERS virus in secretion and milk, and they develop antibody. Transmission is from camels to human being. Spread to 7 27 countries and result in 2,279 cases, 806 deaths, which represent 35.4% mortality rate. Last cases of MERS was detected in July in 2015 in South Korea and China, and the seventh virus, which is the new virus SARS-CoV-2, which is the novel coronavirus that causes the new disease COVID-19, which discovered in Wuhan, China in December 31, 2019. And now there is about 3,909,050, 320 cases, and deaths, which is 2071, 791 there, and death will be mortality rate 6.88. Spread to 2012, 212 countries, and they believe that all SARS, MERS, and SARS-2 genome sequences found closely related to the bad coronaviruses. Uh, Animal coronaviruses infect all most of animals from dogs, cats, horse, cattle, swine, chicken, turkey, rat, mouse, and rabbit. And also there is uh, rabbit, and the bat viruses belongs to both. Most important for us, you remember calf diarrhea virus, which causes diarrhea in newborn calf, and bovine respiratory disease. And in chicken, we have the IB, which is infectious bronchitis virus, which causes severe respiratory illness in chicken. My student Abad discovered or isolated this virus. Um, the genome and the origin of SARS-CoV-2, it is a new virus, was identified and isolated, uh, and its RNA was sequenced in January 7, 2020. They found that 95% homology with the banned virus and 75% with SARS virus. And this sequence of the virus had been distributed to many central labs in the world for diagnosis and to do research on it in the central bank. Viral RNA is not segmented. Here you see uh, the genome or the RNA of COVID-19. You'll see has been distributed by Wuhan, and they put it in the gene bank, and you see there is 30,000 base pair. From the right side, you will see the ORF1A, which is the first genes which code for non-structural proteins. Mainly, it is enzymes needed for replication, RNA polymerase. And on the right side, you will see the genes which code for the structural protein, like the spike, like the membrane, like the envelope, and for the nucleocapsid. Origin, want to go to study the origin of this SARS-CoV-19. I believe, and most of the scientists now believe, that it is animal origin. Why? Because the first cases of COVID-19 were discovered in the seafood market in the worker, in that seafood market in Wuhan, in which there was many living and dead animals on meat, mainly for bat, fish, chicken, snake, frog, pig, civet, cats, and others. Later on, cases has been discovered in person not have no connection to the seafood market, and this indicates that human to human transmission. SARS-CoV-2 is a zoonotic virus like SARS and like MERS. Bats, virus, or viruses are the ancestor, and bats are a reservoir of all zoonotic coronaviruses like SARS and MERS. Why? We say it could be what happened in the SARS-CoV-2. It could be a genetic recombination event happened, change the S glycoprotein of the SARS-CoV-2, and the new virus is a mixture of that SARS coronavirus and other unknown beta coronavirus. It could be a recombination. This is a recombination effect at the S uh, glycoprotein at the receptor binding domain region of the ACET receptor. This recombination enhances the transmission and ability of the virus to human. So this means cross species. When this happens, when like bats they get infected with two viruses, there is an exchange of two of genes between the two viruses. Beside that number two, there will be a mutation in the spike S protein may have significantly enhanced more binding and infectivity of virulence of a mutated virus. Mutation, it is a minor change, only few amino acids at the combining site of the S protein. New studies says that pangolins, this animal, يأكل النمل أو pangol, like bats, are reservoir or intermediate host of the SARS-CoV-2. So this is increased the zoonotic potential of the virus. Pangolin coronavirus are 91 identical to SARS-CoV-2 and closer relationship to bat virus, suggesting that pangolin passed the virus to human. And beside that, SARS-CoV-2 is 19 amino acid different from bat virus, but 5 amino acid from the pangolin virus in the S protein, which means that is closer to SARS-CoV-2. Recent studies found about the SARS-CoV, three variants has been developed by the SARS-CoV-2 by doing sequencing study, and these variants are A, B, and C. The A variant is the ancestor type of bat coronavirus which passed to human in China, and the A and C came from the C came from the ancestor which is spread outside East Asia to Europe and America. B type is the common variant in East Asia after a mutation and spread outside, even though outside East Asia. Mutated A were seen in America, came from China, and type C variant, major European type, found in the early patients from France, Italy, Sweden, and England. Variant A is the most closely related virus to bat and to pangolin and has been changed to B due to two mutations, and then the B changed to C also by mutation. Recent research, this is a few days ago, say that 14 mutations happened to the virus in USA, even in the S gene from March, with increased virulence. Means the virus, you'll see it in the United States, is more contagious, more virulent than the original virus. Official in USA and Europe says that SARS-CoV-2 leaked from the Chinese Central Virology Lab as an accident, and it is not man-made. Scientists, what they say, it came from bat to intermediate to human transmission, or emerged in nature in a person with a special condition like depressed immune response from food market in Wuhan get adopted in that person in him and then passed to others. And this shows the phylogeny which has been put a few days ago. It shows that the COVID, SARS-CoV-2, we see the red circles more than 10. You will see that these are the variants of SARS-CoV-2. And here shows for you the horseshoe bats, which are the reservoir of SARS-CoV-2, 96% identical to the SARS-CoV-2. And here is a pangolin, which is the intermediate horse for SARS-CoV-2. And here shows for you the transmission cycle from the bat to the pangolin to the human, and then through droplets spread to uh, humans, between a human. And now you'll see the dogs, cat, tigers, lions get infected from the diseased human also. Here shows for you the multiplication cycle of corona COVID-2. You'll see after attachment enter the cell and use the cytoplasm as a factory for him. And you'll see the first, after encoding, you will see the viral RNA genome will be translated by ribosomes and then replicate the uh, viral RNA, and then they assemble, the proteins assemble on the rough endoplasmic reticulum and then goes out after completion in a vesicle and get out of the cell. Treatment for COVID-19 drugs has been tested, and there is no evidence till now an ideal antiviral drug, but there is more than 291 clinical trials are going, and the first drug, which is chloroquine and hydroxychloroquine, and this is, you know, that's a malaria and lupus and anti-rheumatoid drug. They believe block viral entry. Lately, in the late April, FDA in USA disapproved the use of this drug due to its toxicity, cardiac toxicity, and retinal. Even the European Union doesn't like to use this drug. The second drug, which is a combination of lopinavir and ritonavir, which is for HIV used and inhibit the protease, but there is a limited trial about this drug. Ribavirin, which is the third antiviral, and you know ribavirin inhibit many RNA viruses, and mainly is used for RSV, which is respiratory syncytia virus, as an inhaler. And this RSV causes severe bronchitis and bronchiolitis in babies and toddlers, and they use this drug for MERS and SARS, also with combination of interferon. Uh, uh, there is no limited activity they found in a treatment of SARS-CoV-2. The fourth drug, which is oseltamivir or Tamiflu, you know it. This is a drug for influenza, which inhibit neuraminidase, and this no documented in vitro activity of effect on SARS-CoV-2. Several current clinical trials to use this oseltamivir showed no role in management, mainly in China, when there is the two conditions, flu and the COVID-19. The fifth drug, which is Finovir, which is Arbidol, or they think that more promising antiviral with unique mechanism of action in targeting the S protein with the ACE receptor, means inhibit membrane fusion or attachment of the virus and entry into the target cell. And this has been approved in China and Russia for influenza first, but also they show that limited trials for COVID showed lower mortality and high discharge from the hospital. The sixth drug, remdesivir, and this is they believe to be the the drug of choice, inhibit many RNA viruses. This corona and flavi, it is the drug for Ebola virus. They used it 2 years ago, currently promising potential therapy for COVID-19. Remdesivir prevent lung hemorrhage and reduced viral lung involvement. Intravenous, they use it intravenous infusion with different doses, and they found they have no evidence of liver and kidney toxicity. Inhibits viral RNA polymerase enzyme. There are many clinical trials are going now in the United States, and also they gave this drug to many European countries for experimental use, but 1 week ago they found NIH, which is the National Institute of Health, they found that this good result, that this remdesivir diminished the time of recovery from 15 to 11 days and diminished the mortality 8 to 11% in over 1,090 patients. And now they are testing in thousands of patients, but still limited supply. That's the thing. The last one, which is number 7, which is favipiravir. This drug inhibits the RNA polymerase enzyme, halting viral replication. It is the drug for Japan flu. They call it Avigan, which is approved in Japan as experimental treatment for COVID-19, and they use it in China, in Wuhan, and they show that it is safe and effective, improve the lung condition for treatment, like Arbidol. Japan gave this drug to 50 countries, some of them in the Middle East, but the first USA trial of favipiravir were recently approved in US to be used in Boston. Anticytokine number 8 now is used in severe cases mainly because of the cytokine interleukin storm, and mainly in severe cases of pneumonia. And they found that it's effective in treatment of such severe COVID pneumonia. And acetamira, which is anti-rheumatic drug, block also the interleukin 6, and they are using many of the immunosuppressive drugs in this storm of cytokines. Immunoglobulin plasma therapy use as convalescent plasma or hyperimmunoglobulin, and first they use that in China, used to treat COVID-19 patients. Many trials in several countries of the world, even in US and in Iraq, as I hear, and the plasma has to be collected from recovered patient at least 14 days before, and COVID should be examined, this plasma should be examined and evaluated before use. Viral vaccines. Vaccines are most important to combat viral disease like COVID-19 is a type of treatment by stimulating the immune response or immune system against infectious diseases. Any type of vaccine has to be for animal or human has to be safe, sterile, potent. Safe when you give it with small or large doses does not cause a disease. Sterile have no pathogen, and potent has to be induce immunity in the vaccinated individual. And mainly for the has to be given the S protein antibody has to. There is about 78 uh, trial of vaccine around the world. Most of them you will see are selected the molecular method of preparation. First vaccine developed was is now working on it is the messenger RNA encapsulated vaccine using nanoparticle, and here the vaccine is prepared encoding a piece of the genetic code. The messenger code, the piece of the for the S protein, and if you use that message in a nanoparticle, that can be injected to the body and synthesize the S protein, and then the body recognize and make that to that protein immune resource or antibody. And this vaccine is developed by Moderna company and has been past the clinical trial. They they tested into 45% with three groups with different doses, and they showed safety and some immune response. Now they're skipping the test from testing animal. They go to the second phase and they use a large number of volunteers to be tested. The company expect this will be experimental vaccine in July of this year. And the second vaccine, which is the adenovector vaccine, and this is to be prepared by Oxford University in UK, and here they selected a weak adenovirus, and they put in the genome is the gene for the mRNA S protein. No, sorry, the gene of the S protein of the adenovirus. And then experimentally they are going to prepare 100 million of these doses at the end, and they test it in ferret as an animal. And similar vaccine is prepared by China also, will be available in December 2020. Other messenger RNA vaccine is prepared by Pfizer in USA, and they are in phase 1, number 2, and the trial has been done in Germany, and now is another trial of the vaccine in United States using 360 uh, volunteer persons. And but the thing which is amazing about this vaccine, they can prepare 4 types of vaccine. This types, each type according to the virus change in the S protein. If there any mutation, they can include. So now they are making this and vaccine 10 million doses at the end of 2020, and inactivated SARS-CoV-2 vaccine. They are also prepared by China, and this is uh, not well, or means this vaccine is easy to prepare to inactivate the virus by chemicals, and then they use it. And this is controlled experiment in a human trial. Similar vaccine also another in China. They propagate the virus in viral cells. DNA plasmid vaccine also, this is another recombinant vaccine. They put the gene or the edge gene in a plasmid, and then they get that plasmid with the gene into a bacteria, and the bacteria start to synthesize the proteins needed for the vaccine. And number 6, which is lenti-vector vaccine. Lentiviruses are also weak viruses, and now they want to put the gene uh, uh, inside the genes of the lentivirus, which is the vector, and they use it. This is in China, and this is need 2 years more to get this vaccine. Another spike DNA, which is also plasmid vaccine, is prepared in Canada, and it will be available by December 2021. Subunit vaccine, which use nanoparticle, and this very amazing vaccine, they use a microneedle of the subunit protein. Here they get the S protein from the virus and they make it as a vaccine. Similar is by vaccine will be by University of Queensland in Australia, producing viral proteins in cell culture. And the last vaccine, which is Novavax vaccine, is meant to create antibodies to cover the spike of the virus. This is to like treatment. So you're giving the hyperimmune sera like in using the horse plasma against the tetanus. So and then the tetanus toxoid, and they use for people infected with tetanus. And this is has been tested in animals, first to human trial, and they use 130 people, patient to treat. It is expected to start to produce in mid-May. Here shows for you the picture how is the synthetic mRNA vaccine developed by Moderna, and FDA is uh, uh, amazing, making good way to say about this vaccine, and this is really how the DNA comes to the messenger RNA, which is code for the F protein and ribosomes. That's really synthetic and to be given to human. Here's شكرا جزيلا لإصغائكم وتحياتي الأستاذ دكتور أنطوان البنا. آسف لطول